Okay, so good afternoon. Normally the topics in this course are clearly definable, more or less, what people are going to talk about, in the sense that it concerns pretty much a disease or a process related to some basic thing. And so the reason why we're having a session on water will become abundantly apparent when you hear the presentations. Normally I write a little something about a week ahead of time and put it on the website, give people a little bit of clue. So instead I kept getting emails saying, what's this water business? What's happening? So we may have lost some of our audience because they didn't get the full information until later. But I assure you, each week there are several hundred people online just here at the NIH. So we have a large audience, no matter what the presentation. Okay, where's the next one? So the ancient mariner, water everywhere, and all the boards to drink, or water everywhere, not a drop to drink. He had a real problem. We have a real problem. And those interested in human health have a real problem many times over when it comes to consideration of water. I think the best lecture I ever heard in my life was given by Joshua Lederberg, who at the time was president of Rockefeller. And Josh talked about the bond angles between the two hydrogens and the oxygen in water. And the point of it was that it's these bond angles that actually give the extraordinary properties to water, which is essential for life in all forms, certainly on Earth. And Josh was arguing that probably quite logically anywhere else that there is life. So we don't have to belabor that water is important, nor that a large part of the Earth's surface, particularly when seen from space, the blue planet, is water, salt water. Fresh water is only about 2-3% of the water on Earth. But water is the only substance that can exist on Earth as sort of a vapor, a solid, and a liquid. And it's the continual transition from those three forms that make it essential for life. For animals, for plants, for protection from cosmic energies and so forth, and insulation, aside from drinking, agriculture, you name it. It's the essence. So when water is threatened, that's pretty serious business. But it's not something that people think about every day in the week because, you know, water is water. Well, how safe is the water that we drink? And how really does anybody know? Does anybody measure? What do they measure when they talk about safety of water? And water hasn't always been so safe. And so today's presentations cover two parts of a giant seesaw. So the first is when water is the medium for the transmission of infectious diseases. Now, these are, many of them, infectious diseases that set the history of mankind because they were great plagues, cholera, and so forth. So the point of the first presentation by Karen Frank is that not only are these ancient diseases of epidemic proportion still with us, particularly in that large part of the world we refer to as the underdeveloped or third world, fourth world, or whatnot. And these things are lethal, and they've developed ways of getting around some of our even public health and certainly pharmacologic means of therapy. So our first speaker, Karen Frank, is going to discuss this aspect. Water in today's world with today's methodology, how do we handle these serious, old, and in some cases emerging almost, infections? Some of them. There are a large number of them. So 30 minutes, Karen can't cover everything. Now, she's a graduate of the University of Pennsylvania, where she got her MD and PhD, trained in pathology in Boston at the Brigham, did research, training at Children's, and then for 12 years was in microbiology at the University of Chicago. And then she came here in 2012, and she's the head of the microbiology clinical diagnostic experimental laboratory in the clinical center. And she's therefore been on the front line of seeing all of the incredible problems in infectious disease that the clinical center is confronted. So the second part of the program concerns an entirely different discipline. And it's a question that deals with why do we have two-headed fish in the Potomac River? How safe is our water? When you buy bottled water, are you really getting a bargain? And is the stuff that's in the water, what is it? And how do you measure it? And so what you will see and learn is that the new technologies, which one might never have dreamed to be a priori applicable to water, turn out to be open enormous opportunities for answering some of these questions. And so Gordon Hager, who is the director of the NIH Center of Excellence in Chromatin Biology, Gordon took his PhD at Washington University, trained as a postdoc in Geneva, and with Bill Rudder in San Francisco, came to the NIH and has been here for quite a while and has an enormous global reputation in terms of the exciting dimensions of chromatin biology. How is chromatin biology tied into water? Listen and you'll hear. So the first speaker is Karen Frank.
Thank you. So good afternoon. I'm very pleased to be participating in this lecture series. As mentioned today, I'm going to speak about waterborne diseases. All right, we'll use this one. So infectious diseases that are transmitted by water, usually with contaminated with fecal material, involve bacteria, viruses, protozoa, and parasitic worms. So I've listed a number of different organisms here. It's not a complete list, but it gives you a range of the diversity of organisms that are involved in waterborne diseases. The organisms highlighted in red, Vibrio, Legionella, Polio, Norovirus, and Schistosoma are the few that I'm going to speak about, giving you a little bit of the history and what kind of diseases they cause, and a little bit of what is the current state of how we treat this disease, or what is the current research involved. So first, the big picture. So diarrheal diseases. There are 4 billion cases of diarrhea per year in the world, and we don't really understand 4 billion. It's a very big number. 2 million deaths per year. That's also a very big number. 90% of these are in children under the age of 5. If you look at the global burden of disease as measured by things like just as the World Health Organization, 4% are due to waterborne infections, so very high percentage. And of those waterborne infections, 88% are due to unsafe water supply, sanitation, and hygiene. So theoretically, fixable problems. And if you look at the picture of a little girl drinking water out of a tin can she got in the puddle, you can imagine what's in that water. Okay, again, mentioning that what we need are critical sanitation facilities. There's a very striking picture of many people in India gathered around a well getting their water. Very striking picture, and a number of the websites that either raise money for these kinds of things mention that women spend 200 million hours per day collecting water. So a huge part of the global population spends their time just getting enough water to survive. Okay, now a lot of what I'll talk about is in the developing world. That's where the biggest burden of disease is, but we also have waterborne diseases in the developed world in the United States. One example would be in travelers. So in 2007, 30 million Americans traveled to the developing world. Up to 50% of those people had diarrhea. About 8% of the people required medical care, and of the people who went for medical care, a quarter of that were to GI symptoms. Of the significant pathogens, the highest percentage was parasites, then bacterial, then viral. So all of the different kinds of organisms playing a role in both travelers and the developing world. So the first of the 5 things I'm going to talk about is polio. So you see in the top right a picture of some children who are very disabled by polio. Polio generally affects children under the age of 5. And in the wild-type infection, 1 in 200 infections would lead to this irreversible paralysis. Of those paralyzed, 5 to 10% would die to the paralysis of their breathing muscles. The others might have paralysis of their legs, for example. One striking feature about polio, because we don't see polio in the United States, is that the number of cases have decreased by 99% since 1988. It's very striking. There were 350 cases in 1998, but only 223 cases in 2012. And the 3 countries that still have endemic polio are Afghanistan, Nigeria, and Pakistan. Okay, what is polio? What does polio cause? So most people get polio don't feel sick. Some number of people will have a viral syndrome. So they'll have fever, tiredness, nausea, headache, sore throat, stiffness. But most people recover completely. So you had a viral syndrome. However, some of them will have this paralysis more often of the legs, or they may die from it. It is spread by person-to-person contact or by eating and drinking things that are contaminated with feces, such as water. So a waterborne disease. Here's a very striking picture that I would say many people in the audience have never seen in person. This is something you've seen in the history books. These are iron lungs that are used to treat people with polio. I mean, it's a very striking picture. And in 1955, there were about 29,000 cases of polio in the United States. However, then we have some vaccines. So Salk developed a vaccine that was very good. The vaccine was made by heating the virus in formalin, which killed them and made them safe for injection. There is another vaccine. It's the Sabin vaccine. So this was developed in 1961 by Sabin, and this is a virus which is an attenuated virus. So you'll see down at the bottom there are 3 serotypes of polio, 1, 2, and 3. An infection with one does not give you resistance or protection to the others. So this is a trivalent vaccine, and you can see by the picture that what they did is they grew the virus in suboptimal conditions in different kinds of cell lines to make it less virulent. And then they would passage it through primates to test for virulence. So then and this is the virus vaccine that's used predominantly in the world, oral vaccine. Now, when I was preparing for this lecture, it was very interesting because I learned a bunch of things I didn't know. And I came across this news article, which was just striking to me. This is a woman who was 5 years old in 1953, and that is a picture of her in an iron lung. The woman is now 65 years old, and this is a picture of her in her iron lung in her home. I did not know that people were still in iron lungs. So there are about 6 to 8 survivors in the U.S. who use iron lungs. So she keeps this going with spare parts in her room. They patch it up. It's an old machine. She can get out of this. She can walk around, but she found that the more sophisticated systems that they have for forcing air in her lung was not compatible with the inflammation she has in her lung, and she finds that she breathes easier with it. So she'd rather breathe with this than the other things, and I just was struck by that. And her concern in making that news article was no one remembers polio, and she's a spokesperson for getting your vaccinations. Okay, another thing that is in the news, which is very striking, is polio in Syria. So due to the war in Syria, we have an outbreak, and it's the first one in 15 years. So Syria had been a country without polio. In December, there were 24 cases reported, and now we have 7 countries having a massive campaign to try and hit 22 million children to try and stem this. And the CDC is currently recommending, if you are an adult traveling to one of the healthcare facilities or refugee camps in Egypt, Iraq, Jordan, Lebanon, or Turkey, that you should get a booster shot to stop the spread. So polio is not gone. Okay, now another thing that is striking about polio, so we had this massive campaign, greatly successful, reduced the polio cases, but now we have an issue of balancing the pros and cons of the vaccine versus a wild-type disease. So oral polio is given a mixture of 3 attenuated viruses, and on occasion, the vaccine can cause poliomyelitis. So the number of cases is 1 in 750,000 cases of paralysis due to the vaccine, and that causes 250 to 500 annually. So that's a big number compared to 0, but it's a little number compared to the number of wild-type cases we had when it was, polio was everywhere. So when polio, you look at the wild type, it's 1 in 100 to 1,000 or 1 in 200. So the numbers are very different in comparison, but when you're giving a healthy child a vaccine and then they get paralysis, that's very striking, very hard. The other thing you can happen besides getting the paralysis directly from the vaccine is you can have a circulating strain derived from the vaccine. So it's not a vaccinated child, it's someone who got the virus because it's circulating. So in the past 12 years, we have 650 cases due to circulating strains. So if we are able to eradicate polio from the world, as we believe we have with smallpox, we may have reemergence because the wild type is not really gone like we think it is, or because we're continuing to give vaccine strains and the vaccine strains are circulating. So what's the current state? What is the research that's going on, the new strategies for polio? And I will add that these strategies have a factor of they're now more complicated and they cost more. So up on the upper left, you see a picture of the polio viruses, serotypes 1, 2, and 3, and you see an extra, the 2. So 2 hasn't been seen. So it's seen to be perhaps eliminated, and 3 hasn't been seen as, is it really eliminated or not? We're questioning, we haven't seen it in terms of the wild type virus. So 1 is the one we're seeing the most for wild type. So some of the recommendations are going from this trivalent vaccine to a mono or bivalent vaccine to get better immunity. So if you don't need all 3, don't give all 3 and get better immunity with 1 or 2, and they're shortening the interval. So my point of this slide is that for us, we've been doing this for years, it's old hat, but people are still working on what's the best strategy for vaccination. The other thing is phasing out the current vaccine by 2016, and then understanding why in the developing world the vaccine is not as effective. It's not as immunogenic. Is that related to the overall health of the children? Is it nutritional state? Is it something more complicated with the immune system? To understand better the emergence of these vaccine strains. Can we do anything about that? Can we make it attenuated in a different way? Working on a better vaccine, and also they're expanding the age of who they're vaccinating because in some of the outbreaks they're seeing older children getting the disease. And the last thing is they're adding an intradermal administration in addition to the oral vaccine to boost the different kinds of immunity you get. And that hasn't been something that's typically seen, but you can see where cost can be added. If you're adding number of times, if you're adding a different kind of injection, can you now do it out in the house or do you have to bring them into the clinic? There's logistical issues to changing your vaccine strategy when you're trying to hit 22 million children in a developing world. Okay, one last thing you might have heard in the news is there was a report of some cases of polio-like disease in California, about 25 cases. And it wasn't polio. They were able to show that there was no evidence of the polio, and there was speculation. Is this enterovirus 68, 71? There are other enteroviruses in this family that can also cause paralysis, and that's a little picture of a girl who was affected with her father carrying her that was in the news. A public health official has said that the number of flaccid paralysis cases in California is actually not more than we see typically in non-endemic, non-polio endemic areas. So it caused a flurry in the news. Public health is saying, well, it's not really out of line, but it shows us that paralysis due to viruses is here. There is some level of that in the United States, and there's more to understand about these viruses. Okay, we're going to move to the second case. We're now going to talk about cholera, Vibrio cholera. It's a gram-negative bacteria you see up in the stain up in the upper left. We are currently in a global pandemic of cholera, which has been happening since 1960. So it started in 1960, and it's still going on. It hasn't stopped. This is the 7th pandemic since the 1800s. It's due to Vibrio cholera, serotype 01L4, specific type. And cholera causes profuse watery diarrhea and can lead to dehydration and death. In 2011, there were 58 countries on 6 continents that had cholera. So it's a widespread problem. 3 to 5 million cases per year, 100 to 200,000 deaths per year. And the little cartoon is a picture of a water pump spewing out cholera. This is from in London when they identified cases surrounding particular streets where they had pumps on the corner and were able to identify particular pumps that were contaminated compared to the more wealthy areas that were not having cholera cases. And they removed the handle from the pump in one case to stop people from using that pump. Okay, what you would have heard most in the news would have been the case in Haiti after the earthquake. So 2010 to 2012, greater than 700,000 cases greater than 8,400 deaths. So huge number of deaths in these poor people in Haiti. And the cause was associated with peacekeepers who came in from Nepal to work to help with the earthquake, and they were on a tributary next to this river, and it was shown that it was a South Asian strain of the Vibrio that went into the water, and this was combined with tens of thousands of people bathing, swimming, drinking in that water. So it was a confluence of multiple factors. The population lacked the immunity because they hadn't had cholera. And then people fled these areas and then dispersed the disease across the country, as well as the Vibrio cholera having a higher production of the toxin. So you had multiple factors. It wasn't one thing. It was like 6 or 7 things that contributed to having such a devastating effect in Haiti. How do you treat cholera? You must give oral rehydration. So even countries that don't have a sophisticated method can add a certain amount of sugar, a certain amount of salt to the water, and feed it orally to the people. So 80 to 90% of the people in an epidemic can be treated with this oral rehydration if you have someone there to give it to them. The other thing is if you have someone with sari cloth, they can filter the water. That's something accessible to them, and it actually gets rid of 99% of the cholera because it's attached to the phytoplankton and particulates. So it gets the cholera out, it reduces the burden of the cholera. So what are current recommendations for treatment? So the thing that is changing, there's been a debate, are antibiotics worth giving? Do we want to give antibiotics that don't make a difference when we can just be giving oral rehydration? Are we just causing resistance unnecessarily? The current recommendation is yes, you are going to give antibiotics if you've got someone who's moderately or severely dehydrated in addition to the aggressive hydration. And now zinc is recommended as part. It's not just sugar, salt. You're going to add zinc into there, and it's going to help with the therapy. Okay, there is evidence to suggest that cholera is increasing, and there are speculations that it may be associated with global warming. So up in the top, you see where we have data, and then I'll show you what the speculation is. So up in the top, they're showing you the North Atlantic Ocean and how much plankton there is in there. And the green is showing an increase in the warmth, and the blue is showing you a decrease in the cold version. Vibrio is a commensal in fish, shrimp, mollusk, and it's in rivers and coastal waters naturally. So there's multiple Vibrio species beyond Vibrio cholera. And the hypothesis is that this change in the warming may be affecting how much cholera is in the waters that are naturally occurring. That hasn't been proven, but people are working. This is one area of research. How does cholera cause this profuse watery diarrhea? You see a cartoon where you have the bacterium, which produces a toxin, an AB toxin, a multi-subunit, that interacts with the ganglioside receptor, affecting an enzyme that produces cyclic AMP. The cyclic AMP is increased, which causes a release of the salt and ions. And there's little letters missing there, which, I mean, this is just a copied picture, but there's supposed to be salt up at the top there. And then you see that there are places in the picture where they're showing where they might target areas for therapy. And I have another picture that hopefully came out better. Okay, so what are the current recommendations for vaccines for cholera? So should we be giving vaccines or not? There is an injectable vaccine, which is a killed vaccine, that has been used widely in the world. However, when there was a very good study, it showed that the protection was not that great, only 50% protection over 6 months, and it led to a decrease in people thinking the vaccine was worth, and a change in the strategy worldwide. There are newer oral vaccines that have been developed that are believed to confer better immunity, and there was a change in the recommendation by the World Health Organization. So the recommendation being, if you have a highly endemic area of high risk of endemic cholera, you should be vaccinating. And if the outbreak starts, it's worth jumping in there after the reactive deployment. So the epidemic's already here, still go in there and give vaccines, and you're going to change the dynamic of the epidemic. It's not too late. Okay, the next 4 slides are a series of pictures from a recent paper, 2013, showing you a whole series of chemical compounds that are proposed to be used for cholera. So might one of these become a better drug for treating cholera? And then in each one, it has what is the target in those series of steps of how the cholera toxin works. So the first left is on motility and virulence of the toxin, of the bacterium. On the right is inhibitors of the ganglioside receptor in the toxin binding step. Then you have in the center here inhibitors of the protein that refolds in the cytoplasm as is working. If we can inhibit that step. Then you have inhibitors of the cyclic AMP metabolism. So 4 different targets that you're getting. And then the last one is a whole series of protein of molecules to inhibit the cystic fibrosis transmembrane conductance regulator, which was at the top of that cell. I don't know if it wasn't labeled well, where the salt in the water was going through. So these are a series of potential drugs that may be developed to fight cholera. Okay, let's move to a new disease, number 3. This is a different kind of disease. It's a blood fluke or a worm, schistosomiasis. And you can see from the striking pictures that this can be a chronic or an acute infection. So you can have a chronic disease where they live with it, they have anemia, malnutrition, or you could have an acute case where they have abdominal pain, diarrhea, hepatosplenomegaly, in some cases cystitis, it depends on the species of the schistosoma, and in some cases cerebral disease. Schistosomiasis is primarily a tropical and subtropical disease, and it's due to infested water. And it could be agriculture, domestic, recreational activities, different ways that people use the water. And again, a huge burden worldwide. 200 million people affected in 2012 and about 42 million people being treated annually. So huge numbers. Okay, the life cycle of schistosoma is different than, than a bacteria that I was speaking about or a virus. This is a complicated cycle. We start down in the bottom. You have 3 different types of schistosoma. You have the eggs that are hatched to a myricydia that goes into a snail, an intermediate host. It develops into sporocysts in the snails and then is released as a free living form that then penetrates the skin in the human. So that's how it enters the human's body, where it develops and matures into these adult worms. The worms then lay the eggs and start the cycle again. So there are some attempts to control schistosomiasis by having periodic large-scale treatment of populations by praziquantel, and of course improvement of the water, having potable water, complete sanitation, and discussion of possible snail control. I don't know exactly the methods that they would focus on, but the snail being an intermediate, it can be a potential target to address. Okay, what is current research in schistosomiasis? So what I have is not a potential therapy, but it's on the diagnostic assays that you're using to detect schistosomiasis. They say, why do we need that? Someone gets very sick, I can see the symptoms, they've got eggs in their stool, and the answer is because you have places where they're trying to clear it and the assays are not sensitive enough. So looking at the eggs under the microscope is not sensitive enough in an area that has a low endemicity, or they have a neurologic symptoms, and so again, you're not maybe detecting it in the stool. So there are investigators working on different assays. These are immunoassays, such as ELISA, a rapid point-of-care immunoassay, which would be very relevant in a third world where they would need point-of-care testing, and a real-time PCR assay. The real-time PCR assay might be very great, sensitive, and specific. You may or may not be able to use it in the region of the world that you need it in at the moment. And the last immunoassay was changing the antigen that was being detected. It was the circulating cathodic antigen. And I don't know that you can see it, but little picture was showing you red fluorescence that was being used in that assay. So people working to develop better assays for schistosomiasis. And this is the last slide on schistosomiasis, and this is a group that was taking a different approach for detection. They started looking at microRNAs. So these are non-coding RNAs that are, have a number of functions both in humans as well as in microorganisms. And their first attempt was to look at the spectrum of microRNAs in the human who has schistosomiasis. And their idea was, can we follow the biomarkers? And the answer, it was too variable. So even though a panel of people had it, which microRNA was up or down was not reliable enough. However, the 3 shown here were from the worm. And they found that these may be useful tools for diagnosis, that we can detect these 3 microRNAs in the worm, and that may give us better sensitivity. So again, this is a 2014 paper, so very recent. We'll have to see if this pans out. Okay, organism number 4. We're moving back to viruses, and we're going to look at noroviruses. So this is what you would see in the newspapers when you read it, and you can see that in a very short period of time, over a month, you see 4 different cruise liners reporting an outbreak of norovirus. Makes you wonder why people go on these cruises. And what does it cause? So it causes nausea and forceful vomiting, diarrhea, abdominal pain. And I'm showing you a cruise liner, but if you look at the numbers again, 200,000 deaths in the developing world in children under the age of 5. So the cruise liner is what we see in the news, but that's not the highest burden of disease. In the United States, we do see 21 million cases, so it's a lot of cases, not as many deaths. And you'll note one thing, when you have a child in the United States who goes into emergency room with GI symptoms, 21% may have norovirus positivity. But you have to be careful because 4% of the healthy kids are going to have norovirus positive. So there's a chance when you're looking at that child that he happens to have norovirus positivity, but he's got another symptom for his GI. So the clinicians have to be careful and be aware of these when they're diagnosing a child with GI symptoms. But in the United States, we do have 800 deaths, mostly in young children and older adults, so not insignificant, and a huge cost to our health, burden in terms of cost, $2 billion in terms of productivity lost, hospitalization, et cetera. Why is this the case? Why is norovirus so bad? And the answer is it's because it's very stable in the environment. It has a very low infectious dose. It doesn't take much to cause someone to get sick. It's only moderately virulent, but it has a very high load in the feces. And one example of that is there's a report in the literature where there was one baker who contaminated some icing that he was using in his bake shop, and there were 3,000 cases 4 days after that from his cupcakes. Okay, this is a model that has been developed at the Health and Safety Laboratory in the UK to explain what's going on with norovirus. So this is a mannequin that has forceful vomiting, a fluorescent vomit. And what they do is they put this into an area, use the instrument, and then they show with UV light how far the particles have spread. And there have been cases where someone vomits in a restaurant and someone across the restaurant gets norovirus. So it was aerosolized, and they were able to get sick from that. So it's not just drinking, water that had feces in it. Okay, now I've been mentioning cruise liners, et cetera, but in the United States, the burden of disease is in a long-term care facility. So 60% of the norovirus is affecting people who are debilitated or compromised in some way in these long-term care facilities and less able to fight it off. Okay, what are we doing currently with norovirus in terms of diagnostics, research, et cetera? This is a panel showing you a molecular assay, which is one of a series of molecular assays that are being developed and being submitted for FDA approval, et cetera, that test a wide range of GI pathogens. And you can see it includes bacteria, viruses, and parasites all in one multiplex panel, norovirus just being a specific example of that. One thing that has to be considered in the clinical laboratory when you're looking at these things is the cost worth it? Am I getting the sensitivity and specificity in each of these individual microorganisms compared to whatever my other test was that I was using individually? And would I rather do 15 tests or the 2 that I want to do or micropanel? There's decisions to be made. Cost is one of the biggest factors. And the other thing is these assays are actually very hard to validate because if you had one assay, you know what number of samples you need to say you got sensitivity and specificity. If you have 15 targets, you need 15 targets times that many positives. It's much harder to get your validations done. Okay, why don't we have a vaccine for norovirus? And the answer is partly we can't grow it in culture. We don't have a robust animal model. And there's antigenic and genetic drift making designing a target very difficult. So this is analogous to influenza viruses and influenza vaccines. It has the antigenic drift, and the immunity is short-lived. So if someone has the infection with this norovirus, 2 years later they can get the same strain again and they can get sick again. They don't have lifelong immunity. So one approach that researchers have taken to tackle norovirus is to use virus-like particles. So you don't have the virus replicating, but you have different series of, the P dimer, the P particle, the S particle, and you make a virus-like particle. And they have had some success in some models for immunizing with these particles. I think this is the last slide, maybe last slide. What this is showing you is the antigenic drift. So this is highlighting particular epitopes in the norovirus, and these New Orleans and Minerva strains are naturally occurring strains, and they're seeing a change and an escape from herd immunity with a change in an epitope. So if you take your particle and you design it with a particular epitope to have a vaccine, but then the virus shifts and you no longer had the right epitope, you no longer have a good vaccine. So you would probably need a multi-pronged approach with multiple antigens if this were to work. Okay, this is the last organism I'm going to talk about for waterborne infections, and this is back to the United States now. And what you can see from this graph, it's a CDC graph, and you're looking for the hash marks, and what you see is there's more hash marks over in the current years than there is before. The hash marks are Legionella. So we're seeing more Legionella proportionately in terms of the waterborne disease in the United States. In 2009, 2010, we had 45 outbreaks in the United States from drinking water and recreational water, 1,200 cases, and of the deaths due to waterborne disease, 14 of 15 of them were Legionella. So proportionally, it's a problem. What is Legionella? So it's a gram-negative bacterium, causes pneumonia, you can see by the x-ray, and we have particular media that can be used to pick it up in culture. So it causes Legionnaire's disease or Pontiac fever, with 8,000 to 18,000 people hospitalized in the United States. The pneumonia might be very mild all the way to fatal, 12% fatality from Legionella pneumonia. How do we get the name Legionnaire's disease? In 1976, the American Legion had a conference in Philadelphia, and at that conference, 220 people got sick with a pneumonia-like illness. Nobody knew why at the time. 34 of those people died. They discovered it was due to the Legionella organism. Legionella is not transmitted person to person like some of the things I've been talking about. It's transmitted from aerosolized water that contains Legionella. So it's the shower, the hot tub, the decorative fountain. Those are the places that people can get Legionella. Legionella is fascinating. It could be a whole lecture in and of itself in terms of the pathobiology of it. So Legionella in the far left comes into a cell and goes into a vacuole. It's escaping from the endosomes of the cell. It fuses with a mitochondria, and then it uses ribosomal proteins to form a vacuole that it then can replicate inside of, hiding from the immune system in the endosomes. This is a very complicated slide from a review article showing you ways that Legionella is hijacking the host cell. It's not just one thing, it's a whole bunch of different things. So up on the left, you've got interactions with the endoplasmic reticulum proteins. You've got membrane phosphoinositides. You've got ubiquitination, apoptosis, cell stress, protein synthesis. So each one of those, I'm sure there's 3 to 5 labs or 10 labs working on that one pathway. So there's a huge amount of research going in to understand how this intracellular pathogen is working, and can we target some of those? Can we get a better treatment for Legionella if we investigate these? So I'll end with some pictures, striking pictures. So again, these diseases often happen in areas of the world that have a catastrophe, an earthquake, a flood. And this whole issue is as much a societal, political, and engineering issue as it is a medical issue. So for some of the things I talked about, we do understand enough that we can treat it, and we don't see it in the United States, but it hasn't been implemented across the world. A small piece of good news is that in the past 20 years, the number of people without safe drinking water has dropped by 2-fold. So we have a long way to go, but that is progress headed in the right direction. Thank you very much. Any questions?
I didn't quite follow the argument about how polio was being transmitted or recreated, as it were, from the vaccine. So are you suggesting that some virus is surviving the...
Yeah, okay, so these are live vaccines. So the
but they're supposed to be disabled, right?
Well, okay, so if you look, it's very interesting. It's amazing to me that it actually works. So in the Sabin vaccine, they take a virus and they passage it many, many times through these cell culture systems, through a monkey, and then you get this virus. So something happened to the virus to make it less virulent. So it had a mutation in a certain protein, and they can revert their mutation. So that's part of the research. So we know that it's happening. We know that people get disease, and we know that they're circulating virus. So it's not dead. So that is some of the discussion is, Should we go back to a Salk-type vaccine? Is it good enough? You know, when do you have the balance if we have too many vaccine cases compared to wild-type cases that the balance shifts and I no longer want to use a live vaccine? But there is research to understand what is reverting, and I didn't see the answer to that if they know which proteins and mutations are changing.
So I remember when they built the Aswan Dam and the schisto flourished upstream, very little attention was paid to that as a likely possibility. But then I believe that came under control, right? By some, I don't know what they put in the water. How did they handle that?
No, I don't know that one. Sorry. Thank you.
All right, well, thank you very, very much. And so we'll continue with Gordon.
Okay, so we're going to switch gears quite a bit here to something that's much more unknown. That is it a problem or is it not a problem? In the past 2 or 3 decades, there's been increasing concern that because of agricultural runoff, effluent from large industrial firms, pharmaceutical industry, there's possibly compounds coming into our water supplies that are not good for human health. Just a quick example, Wynn mentioned this as introduction. In the upper reaches of the Potomac River, there are places where essentially all the smallmouth bass are transsex. That is, because of estrogen-like activities in the water that they're swimming in, the males have become transsex animals. So this, certainly for fish living in certain places, this is a problem. But the question really is this a problem for humans? And this general phenomenon is referred to as endocrine disruptors. So the WHO refers to an endocrine disruptor as a compound or a mixture of compounds that affects the endocrine system and causes adverse effects in intact organism, its progeny, or in populations. And the characteristics, there are several characteristics of these group of potential disruptors. We can have either low-dose effects or high-dose effects. Particularly low-dose effects at very long-term exposure might be a problem. There's a wide range of effects in the human potential in the human organism. There's lots of signaling pathways that are subjected to these disruptors. Nuclear receptors, membrane receptors are obvious targets for molecules like this, and as we all know, the endocrine system is enormously important in controlling homeostasis in the human adult or in the human animal. There can be persistent effects, particularly in developmental systems, seem to be more sensitive potentially to these kind of compounds, and even potentially there can even be transgenerational effects. So the big question is, in some very specific cases, we know that such chemicals can be present in certain areas of the environment, but how big a problem is it? Particularly, a part of this problem that we're particularly interested in is nuclear receptors. These are enormously important group of signaling molecules in mammalian cells. In humans, there are 48 of these receptors. I've listed a simple diagram of the various families here. And of course, many of these receptors have very high affinities for traditional steroids, traditional hormones that you're all familiar with, estrogens, progestins, et cetera. And since these receptors are have such high affinities for these molecules, it becomes an important question whether activities like this might be present in the environment. If they were, they could have very serious effects. And the reason we got interested in is not because we're water people, but because we use nuclear receptors as models to study gene regulation in animals. And since we have such interesting reagents, we sort of decided a couple of years ago to take a peek at whether we could see these activities in the environment. So how might you go about, what kind of an assay could you use? And really, assays are the big problem in this field. And something you can do that's interesting with nuclear receptors is that some of them glucocorticoid receptor here as an example will actually move around in the cell in a very dramatic way. So the glucocorticoid receptor is typically located in the cytoplasm of the cell. You can see in the top panel of these group of this exposure series. As you increase the concentration of ligand, shown on the left here, the receptor increasingly moves to the nucleus. So you essentially have a total movement from a cytoplasmic localization to a nuclear localization. So when you label these molecules with fluorescent tags, such as the green fluorescent protein or other fluorescent tags, you can very easily monitor the movement of these receptors and thereby detect the presence of an activity that acts on that molecule with a living cell assay. And this is the advantage of this approach is that of course, it's very fast, it's very simple. All you have to do is drop something on the cells, and if there's significant concentration of an activity that acts on that molecule, you'll see it move to the nucleus. And other receptors will also behave in a similar way. This is an example of the Aryl hydrocarbon receptor, for example, which some of you may remember this event in the Love Canal back in 76 and 77, where it was discovered there was very serious contamination of the local environment in that community by this terrible chemical called dioxin, which is a byproduct of some of the industrial processes. And again, in this particular case, we can see this receptor, when it's labeled with a fluorescent tag, is completely in the cytoplasm, or almost completely, and you get a dramatic movement to the nucleus when you activate that receptor with dioxin. So this is, again, an example where you can actually see the presence or absence of this activity in an environment. Here's another example. This is the androgen receptor. Androgen receptor is not quite so dramatically localized in the absence of its ligand. There's some in the nucleus, but still a lot of it in the cytoplasm, and you get a complete sequestration in the nucleus when it's activated with one of its ligands. So these are examples of how we could use this to see activities. And progesterone receptor is another example. But what about the receptors that don't translocate? Remember, these are nuclear receptors, and they're called nuclear receptors because they work in the nucleus, mostly, not uniquely, but mostly they act by modulating gene expression. But they also happen to be mostly in the nucleus. So most of these receptors, you look at them in the unliganded state, they're already in the nucleus. So you can't, how could you possibly use this kind of assay to look at those activities? It turns out there's a way to do that, and this is something we developed a few years ago. So the steroid nuclear receptors have a very highly conserved domain structure throughout the family, the superfamilies, and they have a C-terminus that's highly conserved, and it's where the hormone, the actual part of the protein that the hormone binds to, called the ligand binding domain. In the middle of the molecule, you have a DNA binding domain. This is where the molecule binds to genes and regulates their expression. And you have this very highly variable N-terminus, which also has some activities that are responsible for activating the gene regulatory properties of the protein. But in some cases, the N-terminal is almost completely missing. And for the vitamin D receptor, for example, there's essentially no N-terminal domain. Now, what happens when the protein binds a steroid, you have this domain that I described as being so conserved is actually a 12-helix bundle, which is highly conserved throughout the family. When the hormone binds in the binding pocket of this bundle, this part of the protein undergoes a conformational shift, a so-called flip of the helix 12, and now it becomes an activated receptor. And since this is such a conserved domain, you can think about possibly using this as a way to create chimeric receptors that themselves will translocate. And this is something we did a few years ago. So we take the ligand binding domain of a receptor like retinoic acid receptor, which it does not translocate. We fuse it into the DNA. We replace the LBD of the glucocorticoid receptor with with the RAR LBD. Now you can create a molecule that potentially would behave like a glucocorticoid receptor, even though it would bind retinoic acid as its ligand. And in fact, that works. So here's an example of one of these fusion chimeras. At the top here, we have GR, the fluorescent labeled GR receptor, completely in the cytoplasm. You add a hormone that activates that receptor. It moves to the nucleus, a very dramatic translocation. And a retinoic acid does nothing to the movement of that molecule. But if you take this, and RAR again doesn't respond to the coordinate ligand for glucocorticoid receptor. But if you look at this chimera, now we've got a retinoic acid responding chimera that's in the cytoplasm, doesn't respond to the hormone for glucocorticoid receptor, but does move to the nucleus almost completely in presence of now retinoic acid. So we can take the behavior of the glucocorticoid receptor and transfer it to other members of the receptor family. That gives us now the option of not only looking for activities that are by using the naturally occurring receptors, but using these fusions, we can, in theory, we could extend this activity throughout a large number of the members of the family, which would be very useful. So this is then the general idea of what we'd like to do. We'd like to use these chimeric fusions to use as a general probe for not just corticosteroids, not just androgens, but many activities that act on the human endocrine system in one fell swoop. That if we could do that, would be a powerful tool. And in fact, we've recently shown that we can make this work for thyroid hormone receptor, which is another important member in the human endocrine system. So we've now, we decided to take a leap in the unknown here a couple of years ago and see if we could actually use these tools to detect the presence of these activities in U.S. water supplies or U.S. rivers. And this was work that was initiated in our lab by Diana Stavreva, who's here, and she's got a lot of help from Luba Vardakovsky, as also Lou Schultz is also here. And basically the idea is use these robotic, large-scale screening systems that allow us to see where fluorescently tagged proteins are inside cells in an automated format. So we can now potentially screen very large numbers of samples automatically. And so we applied this approach to a set of water samples that we got from our collaborator, Luka Ivanovitz, and his colleagues at the United States Geologic Survey out here in Western Maryland. And this is just a complicated slide that shows you a lot of informatics that's used to do the screening process. But what you'll immediately recognize is that there are activities popping up in water samples from throughout various places in the northeastern United States. Very significant, easily detectable signals. In fact, here's a map showing spots. These are regions of waterways in the northeastern United States that have either corticosteroid activity, androgen activity, or both. In fact, it was a little stunning when we first saw these. We certainly didn't expect to be able to detect. So what this showed us was that these assays actually work. We're able to detect, using these live cell assays, the activity of these hormones in water systems. That was telling us that there's two important results of that. One is that the systems work. That is, this is a viable way to look for these activities. And secondly, we're actually finding these activities in places we didn't expect. In fact, this is the first paper where we reported this activity from over 100 sites that were studied in 14 states. 27% of the sites contained detectable corticosteroid activity and 35% contained detectable androgen activity. This is the first time these activities had ever been detected in U.S. waterways. So this raises, of course, a serious question. More recently, we've screened another 315 samples from different sites. Again, samples that were supplied to us by Luka Ivanovitz. And once again, we're seeing activities pretty much throughout the regions that we're studying. So a major question is, what are the actual molecules that are active at a given site? And so we set about looking at some of our more significant samples, or samples with one of the highest activities. Can we actually chemically identify the compound that's present in this water site that's causing this receptor to be activated? And here's an example of one sample. This is a mass spec HPLC chromatogram, you can see that there are multiple, and even in this one sample, there are multiple activities that are being picked up as active in the translocation assay. We try to go, we purify those samples. We tried with working with colleagues in Frederick to identify the exact chemical, make a chemical identity of the actual molecule that's responsible for this activity, but it turned out to be a fruitless activity. This is one potential compound that was identified. Here's another one that was identified, but there was never any clear identification made as the exact active molecule in this given sample. And this brings up what is the second major problem in this whole area. And that is, once you throw this stuff in the water, it doesn't really stay very long at what it was when it went into the water, because of course there's enormous chemical, a lot of ultraviolet light in water systems, a lot of oxidation. You're never going to keep one of these complicated steroid-like compounds in its same identity once it's in the water. And this is a big problem. So if you're worried about this problem, are there these activities in the water system? And if so, what are they? You have a tremendous difficulty. This is another complexity. It was illustrated recently by this paper from a couple of guys who were looking at this tribonolone, which is tribonolone acetate, which is a synthetic anabolic steroid that's used as a growth promoter in the U.S. cattle industry. In fact, 20 million cattle per year are treated with this compound. And what these guys discovered was you take the active form of this anabolic steroid, and they subjected it to multiple light and dark cycles in the laboratory, as the compound would see if it was in the water, in a waterway somewhere. And they discovered that during the day it's degraded, but during the night, or during the dark cycle, it reconstitutes itself. That is, it appears again. So a compound that's being degraded under one condition is actually reconstituting, reappearing when you go into the dark cycle. So that's obviously another problem. If you're going to try and figure out what's in the water supply, if it's obviously being converted from one form to another. So that particular paper was reviewed in Nature News and Views recently, and they came up. The important conclusions that they made were, first of all, that there are hormone-disrupting chemicals may be far more prevalent in lakes and rivers than previously thought. And more importantly, these endocrine disruptors, pollutants that unbalance hormone systems, are known to obviously harm fish. Of course, fish are living in the water. They have a potentially much greater exposure. But there's growing evidence that some human health problems might be actually linked to these endocrine disrupting activities in water systems. And again, they point out pinpointing these culprits from the vast array of these chemicals that are present at very low concentration is going to be very difficult. And in fact, one of the central arguments we're making is that it's a fool's errand to try and go into specific regions, specific sites, and identify a specific chemical. It simply cannot be done. And unfortunately, that's the approach that current water monitoring agencies take. They have a list of chemicals that they know are bad, for dioxin, for example, and they can, if they suspect that there's a bad chemical at one particular site, they can look for that chemical specifically, with techniques that are in general very expensive, mass spec, for example. And that's not just going to be possible on any kind of large scale at all. In fact, as I said, it would be a waste of time because you don't exactly know what to look for in most cases. Dioxin is probably an outlier. It happens to be so toxic, it's a tiny concentration that you can easily pick it up and find it. The hormones, for example, are much less, in general, don't have quite such high affinities for the receptors, and they're being turned into compounds that we don't even know. So how are you going to do any kind of screening program if you don't exactly know what to look for? And our argument is you have to have an activity assay rather than a specific chemical assay. And this is the reason we know so little about the prevalence or the presence of these compounds in the U.S. water system, because nobody knows really how to look for them. So the conclusions from our work are, first of all, there is a potential contamination of waterways with this group of endocrine disrupting chemicals, these hormone receptors, or these hormone-like molecules that act on nuclear receptors, and it's largely unstudied. And the reason it's unstudied is, as I've said, there's really not, prior to the appearance of these kind of activity assays, there's really been no way to do it. So it's kind of a big black box. If you look in very specific places with very specific assays, you can find stuff, and the question becomes, how widespread is this? And is this a problem in general in U.S. waterway or worldwide waterway system? As I showed you, we can detect levels of corticosteroid and androgen activities. These are the only two that we've screened for so far, but of course, now we will be able to screen for several others. And when we look carefully, we find presence of these compounds with surprising frequency. And that's, of course, disturbing. The key question is, are these levels of significance in relation to human health? Do they persist in water that's supplied, that's gone through water purification facilities, and actually show up in your water? And we don't know the answer to that, because again, nobody's really looked. Are there low-level effects? Even if they're not concentrations high enough that would really activate a receptor in a given biological exposure, if you're exposed to these even to lower levels that you would not think would activate the receptors over a very long period of time, are there low-level effects that might occur after long times of exposure? And this is a problem that people worry about. And as I said, screening assays that look for specific chemicals, that is, a given specific known chemical that might activate a given receptor, are basically of little value in addressing this question, because these compounds don't stay the way they are once they're out in the wild, in the environment. And finally, as I showed you, the rapid conversion of complex molecules to many forms in an environment indicates that activity assays have to be developed in order to carry out a significant screening program throughout the environment of our country and other countries. And this raises, in turn, a regulatory issue. Those of you who are at all familiar with regulatory processes realize that you can't write a regulation about something that's unknown. You have to say, well, if we know dioxin's bad, so we can write a regulation that says you have to keep dioxin levels at some extremely low level in your effluent from your process, whatever it is, or else it's not legal. But as soon as you talk about an activity, but you don't know what the chemical form of the activity is, there are going to be regulatory problems of trying to write regulation that would in any way relate to controlling that activity. But that doesn't mean it's not important. And we're not arguing that there's a trouble in the U.S. water system. That is not our argument, because we don't know. Our argument is that we know so little about this. We need to have large-scale screens that tell us where, on a countrywide basis, not just these few isolated sites we've looked at, is this a significant problem? Is there a lot of this stuff floating around in general in the U.S. water system? And importantly, as you run the water through the treatment plants, do these activities disappear? And we simply don't know the answer to that. So our argument is we now have tools. There is potentially, we have evidence that these compounds exist at frequencies that we would never have guessed. We have no idea where they're coming from. Of course, the immediate suspects are always agricultural runoff and industrial effluent. But there's very little evidence to tell us exactly where they're coming from. So our argument is we need to have large-scale screens that we can apply in an inexpensive, robust way throughout the water systems of the United States and find out once and for all, how much of this stuff is out there. And that's our story. I'll be happy to take any questions.
Thank you. Yeah.
So you showed us your original data where you were detecting this in particular sites and you got the water from someone. And then you mentioned later, I think, that you don't know if it's in the water that's gone through the...
Right.
So have you taken any samples out of a tap in any of those? Are you planning to?
Well, we're trying to develop support. We've got our focus in the past, this is not We're a basic science lab that studies how receptors regulate gene expression and cause cancer, et cetera, et cetera. That's been we stumbled into this more or less by accident. And we have these, our basic argument is we have these reagents that could be used on a large scale. But now once you start moving from a few sites, a few hotspots to some kind of large scale screen and doing the follow-up studies you mentioned, it becomes a significant, support issue.
Were the samples that were given to you suspicious for some other reason?
Not really. I mean, these are samples that have been collected by the U.S. Geologic Survey for over a period of years. Some of these samples are quite old.
Are there any human studies that show that certain basins, water basins, produce certain diseases that might be related to some of the findings that you have?
Well, there's obvious examples in the Love Canal, for example, a tremendous occurrence of cancer.
Yeah.
But on a large, are there any? There's a question of whether hormones, in particular, have been associated with specific, local outbreaks of increased level of cancer or other types of diseases. I don't know that anybody's ever looked at that because they don't really know what hormones are there.
Another question I have is all this business of buying bottled water and filters for your sink and all that kind of stuff, does that overcome some of these issues or have you tested for that?
If you run your water through a charcoal filter, many of these commercial filters are charcoal basically, you're going to remove a lot of these steroids, that's for sure. But one of the issues we're trying to develop is to get some commercial interest in screening systems that will allow people to screen their own water and say, okay, we can tell you that your water is okay. And the problem we've run into immediately is the commercial people will say, well, the American public don't know that there's any hormones in the water. So we can't sell them anything they're going to buy because they don't even know it's there. So it's catch-22.
Yeah, but people go around talking and say they can't drink the regular drinking water, they have to buy bottled water, and I don't understand whether there's a real rationale for that.
Well, there's a lot of superstition and,
What?
Well, I think there's a lot of superstition involved in those kinds of attitudes. Yeah.
No other questions? So, Gordon, a few years ago, in fact, I guess it's probably almost 15 now, there were several groups that were looking at the decline in male sperm counts. And they tried epidemiologically to relate that to these plasticizers,
Right.
which have estrogen-like activity.
Right.
So I don't know whether, they probably didn't have water samples in those studies, but what's your take on that kind of study?
Well, I think I don't have an opinion. I mean, most of these studies would fall in the category of provocative.
Yeah.
There are very little solid science that says this is bad and this is good. And again, the main reason for this is we have so little information. If you just ignore that there might be a problem there, then obviously you're never going to have to worry about it. But I think there's enough preliminary evidence to say that there should be some effort to figure out what the level of, if it's a concentration, there's a concentration problem. Is there enough concentration of some of these compounds at certain places or at a significant number of places that we need to worry about it? My argument is here, we need to reassure ourselves that there is no problem rather than to start, try to show there is a problem. We're not trying to show there's a problem. We're trying to reassure the community that there is no problem. But nobody knows because there's so little that's been done.
To the fish, there's a problem.
Well, for the fish, obviously. But we're not fish, right? We don't swim in the water. At least not for very long.
Any other questions? Okay, well, listen, thank you both. This is very informative.